Thank you, Lonnie. Claudia Buβ is a professor at the Institute of Medical Psychology at Charité University Medicine in Berlin, Germany. She's also an associate professor at the University of California, Irvine School of Medicine in the Department of Pediatrics, Division of Developmental and Behavioral Pediatrics. She received her PhD in psychobiology at the University of Trier and conducted her doctoral and postdoctoral studies focusing on early life stress and brain development at McGill University and the University of California, Irvine. Her research addresses the effects of stress and stress-related biological and behavioral processes during human pregnancy on fetal brain development, as well as other health-related infant outcomes, like body composition, metabolic function, and obesity risk. Her recent work has specifically advanced our understanding of the relationship between maternal placental fetal stress biology and fetal infant neurodevelopmental trajectories by applying multimodal neuroimaging techniques. She is the PI of several NIH-funded and European-based studies that focus on the mechanisms underlying developmental programming of the brain development and the transmission of maternal preconceptual adversity, as well as stress during pregnancy to her child.
All right. So you said that we could handle it either way, to ask questions in the meantime or at the end. I think if there are any questions that make it difficult for you to understand what I'm saying, if I left out any important details, please feel free to interrupt. Other than that, I think it would be a little easier to have the discussion at the end, but we can do either. So if you cannot wait with your question, just interrupt me. I will not be able to see you, so you'll just have to speak. Okay. All right. So thanks again. I'll be talking about early life stress and brain development and implications for cognitive function and mental health. And the majority of work that I will be presenting today is work that I have been conducting during my time when I was still full-time at the University of California, Irvine, because this is where we have built several prospective longitudinal studies of mothers and their children, and we have been continuously following these children up. We also, in the last few years, have established additional new cohorts in Germany, but also in the context of the large ECHO program in Pittsburgh and at Rochester, but these data are not quite ready yet. We're still in the process of collecting this data. So really, most of this work that I'm presenting today is from studies we have conducted in Irvine. Okay. So I think I don't really have to introduce this general field of fetal developmental programming of health and disease to this group, but it's certainly a topic that is not only studied very heavily in the scientific community, but it's also a topic that always receives a lot of attention in the public media, as you can see here. Because this concept, this idea that something that the fetus, the embryo experiences so early on in life will kind of set the stage and affect disease susceptibility across the lifespan is really intriguing. And we really do want to understand the mechanisms better to help the individual develop in a healthy manner and support healthy development. This research field is really very heavily influenced by epidemiological work from David Barker and colleagues, who have shown that birth phenotypes, so small for a low birth weight and small for gestational age and preterm birth, is associated with a lot of later health outcomes like hypertension, coronary artery disease, diabetes, endocrine cancers, pulmonary function, but also psychopathology. And while there's so much evidence for these associations, it's not believed that it is these adverse birth phenotypes themselves that cause risk for these disorders, but it's rather the conditions during pregnancy, during embryonic and fetal life that affects adverse birth outcomes. So it's more like an intermediary phenotype and an indicator of these adverse conditions during pregnancy that really during the critical and rapid period of embryonic and fetal development can change organ development in a way to predispose individuals for these disorders. And among those many conditions that have been studied during pregnancy that can affect fetal development is maternal obesity, nutrition, infection, environmental toxins, but also stress. And this is specifically what I'm interested in, how maternal stress during pregnancy and even stress that she experienced long before she became pregnant in her own childhood might affect pregnancy physiology in a way that alters the trajectory of embryonic and fetal development and increases risk for psychopathology in later life. So when we talk about programming, it's really the process by which the early environment interacts with genetic and other factors to produce an individual human constitution. And the key assumption of the programming hypothesis is that biological systems undergoing rapid developmental changes are especially vulnerable to organizing and disorganizing influences. So if we keep this key assumption in mind, there's no other organ that develops as rapidly and over such a protracted period of time as the developing brain. And just so that we get an idea, the whole brain, the mature brain with 100 billion neurons and 100 trillion connections evolves from the just 3 millimeter long neural tube. And on average, there are 250,000 neurons that are being generated per minute during gestation. This process is then followed by proliferation of the cells, then migration to where they will stay lifelong in the brain, differentiation, synaptogenesis, and neural pruning. And these processes here are processes that, especially synaptogenesis, is something that is started in late pregnancy, but that continues long in the postnatal period. And neural pruning is mainly taking place in the postnatal period and continues until puberty. So the brain really is highly plastic during embryonic and fetal brain development, but it also remains very plastic in the postnatal period. And this is why when we want to understand the consequences of prenatal exposures, we need to try to differentiate between prenatal and postnatal exposures. And this is why we, in our current studies, we try to study the neonatal brain. So the neonatal brain as kind of an early set point of the brain that has already been influenced by prenatal exposures, but not yet by postnatal exposures. And we try to study then the role of postnatal exposures in addition to these prenatal exposures. Just as a little bit of a framework, so we're interested in how various pre- and postnatal perturbations can affect risk for psychopathology. What are the underlying alterations in the brain in terms of anatomy, but also structural and functional connectivity, to gain a better understanding of how the vulnerability for psychiatric disorders might be shaped in early life, of course, in interaction with the genotype, with a specific genotype, but we're specifically interested in variation in the early environment. What I will be talking about today in my presentation is I will be presenting all human work from our prospective longitudinal studies, showing how our evidence, how maternal stress during pregnancy is associated with child neurodevelopment. Then I will be talking about two very important biological mediators that might underlie associations between maternal stress during pregnancy and child neurodevelopment. We'll be talking about variation in maternal cortisol concentrations during pregnancy and interleukin-6 as one marker of maternal inflammation. And then I will also talk about some of our ongoing studies and some of our data showing that maternal preconceptional stress, her own childhood stress, might also get transmitted to her child via similar biological mechanisms during pregnancy. I will be talking about, I will be presenting data from 2 prospective longitudinal studies that we conducted in Irvine. Here, this study I worked on while I was a postdoc with Kurt Sandman and Pathik Wadhwa. And I came to Irvine when we studied the children when they were between 6 and 9 years of age, and we did MRI assessments in these children and were able to relate these neurophenotypes to pregnancy data that had been collected during these pregnancies. I will also talk about another cohort that we have studied extensively with my colleagues Pathik Wadhwa and Sonja Entringer. I set up these studies.
J'ai un rendez-vous avec le docteur Panton demain à 8h30. Je crois me souvenir que j'avais.
You're fine now. Sorry.
Sorry?
It should be okay. I took, anyways.
Oh.
Someone had unmuted themselves.
Oh, okay. Okay. All right. Sorry. So this is the other cohort that we have established in Irvine and built on with several grants to be able to follow up these children. In this cohort, we have 3 pretty extensive assessments during pregnancy to study maternal stress and stress biology. And then we followed up the offspring starting at birth. And the advantage of this cohort is, as I said earlier, that because we have our first assessment at birth, we can better try to differentiate prenatal from postnatal effects, because at birth we don't assume any postnatal effects yet on the phenotypes we're studying. So when we started this work, there was quite a bit of evidence from epidemiological studies showing that maternal stress during pregnancy was associated with neurodevelopmental and psychiatric disorders, as well as cognitive impairment. But at that point, there were no human studies yet looking at the offspring brains. Everything we knew at that point was really based on preclinical work. And when we studied this, when we published this study, it was the first really showing an association between maternal stress during pregnancy and gray matter volume in these 7-year-old offspring. And here we did voxel-based morphometry, and we looked at gray matter reductions in gray matter volume in association with maternal stress during pregnancy. And what was specifically evident were these reductions in gray matter volume in prefrontal cortical areas, but here also in the superior lateral temporal cortex. So really brain regions that are very important in supporting those cognitive functions that had been shown to be associated with prenatal stress in those epidemiological studies. We then looked further when this sample was larger, and with additional methods, we looked at cortical thickness in these children in association with maternal depression during pregnancy, and again found pretty pervasive reductions in cortical thickness across the brain in association with maternal depressive symptoms during pregnancy. And again, these were especially pronounced here in the prefrontal cortical regions. And it was also the reductions in cortical thickness in these prefrontal regions that mediated the association that we observed between maternal depression during pregnancy and her child's externalizing behaviors. So we then also looked at associations between maternal stress and brain outcomes in our cohort, where we studied the newborn infants. And here specifically, I'm showing you associations between maternal perceived stress during pregnancy and her newborn's hippocampal volume. And you see a negative association. So the higher the stress during pregnancy, the lower the hippocampal volume in the newborns. We are of course always really interested in how much those like variation in neurophenotypes at birth predict later cognitive function and behavioral problems. And what we saw here, which I think is very, very interesting, is that we couldn't see a main effect of the hippocampal volume of the newborns predicting later cognitive function. This was at 6-month age, assessed with the Bayley scales of infant behavior, but that there was an interaction between the size of the hippocampus and environmental enrichment. So this really kind of shows you that there is a cascadic effect, and that like certain phenotypes get established maybe already then in the neonates, but then the next environment, the next exposure is really acting up on this, and it really depends on phenotypes earlier how much of an effect the later environment can have. And what we found here was that it was those infants with a larger hippocampal volume that benefited from the environmental enrichment in terms of this outcome, more than infants with a smaller hippocampal volume. So this, of course, doesn't mean that interventions are not effective in infants who have a smaller hippocampal volume, but for this specific exposure that we tested and this cognitive function, this is really what the data suggested, so that it's not a main effect, but really an interaction with the later environment. We also, in collaboration with our colleagues at the Max Planck Institute and also with colleagues at McGill University, Michael Meany and Kieran O'Donnell, have been looking into changes in DNA methylation in these children. And we've specifically in our newborns looked at a DNA methylation score that has been shown to be different based on glucocorticoid exposure during pregnancy. This is based on this paper that was published in PNAS. So we could show that maternal depression during pregnancy was associated with a different DNA methylation profile and an enrichment of changes in methylation in CpGs that are sensitive to glucocorticoid exposure during pregnancy, and that this methylation score in the newborns was associated with hippocampal volume. So this might potentially be one mechanism how these prenatal exposures could have long-lasting consequences for brain phenotypes. I told you that when we started this work, there were no studies, no MRI studies out there yet on associations between maternal stress and related constructs like depression and anxiety and offspring brain outcomes. And this has really dramatically changed over the last few years. And you see that there is a lot of evidence in fetuses, in newborns, in older children, and also in adolescents and adults, showing associations between maternal stress, depression, anxiety, and brain outcomes based on MRI. The picture is rather heterogeneous because people are looking at different types of stressors and different types of brain outcomes, but there is definitely a pretty clear picture emerging showing that there are associations between maternal stress and brain phenotypes that might increase risk for impaired cognitive function and also increase risk for psychopathology. So we are specifically interested in potential biological mechanisms that might underlie this transmission of the effects of maternal stress to her fetus. And I just want to give you a brief introduction how we think about this. And so just in general, people ask me a lot, so what kind of stress is bad for the mother, and what is the kind of stress that we should be paying attention to, and are certain stressors more detrimental than others? And I think it's very, very difficult to answer this question, because it really depends on how the mother can cope with the stress and how her stress then gets translated into a biological signal. Because if there's no biological signal, meaning that if stress hormones don't increase because of the stress the mother experiences, or this doesn't increase inflammation or oxidative stress and lead to metabolic changes. So if there is no biological signature of maternal stress, then the fetus will not know about the maternal stress. So I always say that the fetus doesn't really care about what kind of stress it is, whether the mother has conflicts with her boss or with her partner, or maybe somebody is really sick in the family. It really all depends on the biological signal, because all communication between mother and fetus is through the placenta and through biological mediators, endocrine and immune biological mediators. One that is very important is cortisol. We know that maternal cortisol can pass the placenta. There is an enzyme, 11-beta-HSD2, that is a partial barrier to the transmission of cortisol across the placenta. About 15% can pass through, which is important because the fetus needs maternal cortisol to develop. So if there was a lack of cortisol, the fetus would not develop appropriately either. But there is a barrier. What is interesting, though, is that it has been shown that this enzyme is also sensitive to stress and is less effective in women who have a high anxiety, for example. So this would mean that not only would there be more cortisol potentially because of the maternal stress, but also of this higher level of cortisol, more would go through because this enzyme is less effective. Then there is CRH in the placenta. CRH, everybody knows from the hypothalamus. Not everybody is aware that there is CRH in the placenta as well in primates. And CRH in the placenta actually has a positive feedback loop with cortisol. So in contrast to hypothalamic CRH. So if cortisol is higher, more CRH gets secreted in the placenta and can then again stimulate the pituitary of the mother and to produce more cortisol. So a positive feedback loop can get initiated, and also higher CRH can then directly affect the fetal adrenals and the fetal HPA axis to produce more cortisol. So this is why if there's higher cortisol in the maternal compartment, we can assume that there's higher concentrations of cortisol in the fetal compartment. And this has been shown in some early studies where they actually paired maternal and fetal blood samples and found a pretty high correlation of above 0.6. Inflammation is also can also be a consequence of maternal stress, but also many other conditions that we are interested in that we know can affect fetal development, like maternal obesity, of course, also infections during pregnancy, like COVID, for example. And one of the inflammatory mediators that we have been studying pretty extensively is interleukin-6. There is some evidence that interleukin-6 can actually pass the placenta and reach the fetus. I don't find that work very convincing. I think it's more likely that maternal inflammation in general leads to placental inflammation, and the placenta itself then secretes cytokines into the fetal compartment. So this is, if we measure inflammatory markers in the maternal compartment, it's probably really just some surrogate markers that indicate general inflammation that then might lead to higher pro-inflammatory cytokines in the fetal compartment as well. So just to summarize, we are really interested in the mechanisms that underlie this association between maternal stress and also other conditions during pregnancy that are associated with cognitive and affective processes and mental health. We believe that maternal placental fetal biology plays a very important role in transmission of maternal stress. Here we have studied, and I will present some data on specifically cortisol and interleukin-6, because we know, especially from preclinical work, that these mediators, while they are very important for normal brain development, if they are present in high concentrations, they can affect the availability of transcription factors, neurotrophic factors, and neurotransmitters, and affect all fundamental processes of brain development, and thereby most likely lead to changes in cognitive and affective processes and affect mental health. So I already talked about why we are interested in glucocorticoids, and we have studied variation in maternal cortisol concentrations in our 2 cohorts. First in our 7-year-old children, where we were able to show that higher maternal cortisol concentrations were associated with larger amygdala volumes in these 7-year-old children. As you can see here, this was only true for girls and it was not true for boys. We could also show that the larger amygdala volumes in girls mediated the association between higher maternal cortisol concentrations and affective symptoms in the offspring. So very early signs of internalizing depressive behavior in these children. When we looked at the association between maternal cortisol concentrations and the newborn amygdala, we saw a tendency for larger amygdala volumes only in girls, but not in boys. Again, the same sex-specific pattern, but it was not quite significant. We then extended the analyses to resting state functional analyses to look at functional connectivity of the amygdala in association with higher maternal cortisol concentrations. And again, we found a sex-specific pattern of association. And specifically, again, in girls, higher cortisol was associated with stronger connectivity to brain regions involved in sensory processing and integration, and very specifically in the supramarginal gyrus and the superior temporal gyrus, as well as the default mode network, so the precuneus and the inferior temporal gyrus. And what was interesting as well is that we could see that this stronger connectivity between the amygdala and the supramarginal gyrus did mediate the association between cortisol and internalizing behavior. So to summarize, we found that elevated maternal cortisol concentrations were associated with larger amygdala volumes that were associated with more affective symptoms and with increased amygdala connectivity with cortical structures that were then associated with higher internalizing problems. And this potentially supports the higher vigilance in offspring of mothers who experience high stress during pregnancy and could increase the risk for affective and anxiety disorders. So whenever we look at any associations between variation in maternal cortisol concentrations, we see sex-specific patterns when we look at neurodevelopmental outcomes. And this is when I move on to the findings, to what we observed in association with variation in maternal inflammation. This is not the case. We see that males and females are equally affected. So such sex-specific programming has been shown in a lot of also preclinical work. There are oftentimes, it seems like male offspring are more affected than female offspring, but it really depends on the kind of exposure and the kind of outcome. Some ideas that we have why this might be the case specifically for cortisol is that we know that there are sex differences in the timing of glucocorticoid receptor expression in the fetal brain. And then also there are sex differences in placental glucocorticoid receptor functioning. And also we know that there are in the mature brain sex difference in the consequences of chronic stress. While we observed dendritic expansion, for example, of medial prefrontal cortex neurons in females, there's retraction in males. So all this could add to the sex-specific effects that we see, but we don't really understand it well yet. want to switch gears a little bit and talk about inflammation now, because I told you why we are really interested in maternal inflammation in the context of maternal stress as well. It is known that the prenatal inflammatory milieu is a risk factor for various neurodevelopmental disorders. This has been established several decades ago, that for example, influenza during pregnancy is a risk factor, and other types of infections is a risk factor for schizophrenia and autism, something that should potentially really worry us now that a lot of pregnant women get exposed to COVID during pregnancy. There is a broad range of gestational conditions that have been identified as risk factors for altered offspring development, like obesity, inflammation and psychological stress, that are all associated with elevated IL-6 concentrations. This is why this is one inflammatory mediator we have picked for our early analyses. We have now also analyzed multiplex assays and a broad range of pro and anti-inflammatory cytokines, and are currently trying to deal with this really large data set that better represents immune processes, but our early work really was focusing on IL-6. We do believe that IL-6 is specifically important for example, this study showed that an interleukin-6 antibody can block effects of maternal inflammation on offspring stress reactivity, social deficits, and learning. So it seems to be specifically about IL-6 as well. I already told you earlier that IL-6 might cross the placenta, but we rather think it's stimulating cytokine production in the placenta. And there's a lot of preclinical evidence for elevated interleukin-6 and maternal immune activation during pregnancy, altering fetal brain development, and some of which we'll hear about today as well. So the interleukin-6 associations we only tested in the cohort where we started testing the children shortly after birth. In the older cohort, we didn't have interleukin-6 during pregnancy. But what we were able to show is that higher maternal interleukin-6 concentrations during pregnancy were associated with larger amygdala volumes. And this was true for males and for females. We also looked at functional connectivity of the amygdala in these neonates in association with higher interleukin-6 during pregnancy, and again found stronger bilateral connectivity to brain regions involved in sensory processing and integration. So the fusiform gyrus, the somatosensory cortex, and also the thalamus. And then also, and what I'm showing here is the stronger connectivity of the amygdala to the salience network, and here specifically the anterior insula. So here you see, just as an example, a scatter plot, how linear this association is between interleukin-6 concentrations and this connectivity between the amygdala and the anterior insula. We then expanded this work as well further, and instead of just focusing on the amygdala, we focused on whole brain functional connectivity and connections between within and between all of these networks that you see here. And again, we saw that higher interleukin-6 during pregnancy was associated with significant changes within networks, specifically again in the salience network that I mentioned earlier, then in the dorsal attention network and then the visual network. But also you see here that there was a lot of associations between higher interleukin-6 and between network connectivity. We also, based on our diffusion data, we did diffusion tensor imaging as well. We specifically were interested in the uncinate fasciculus, which is a white matter pathway that connects the temporal lobe with the inferior frontal gyrus. And we are specifically interested in this in these fibers because we're interested in the frontal limbic connectivity. And what you can see here is the fiber tracks. It's all points along these fiber tracks on the left and on the right. From the frontal cortex to the limbic region. And here you can see in what region of the fiber tract we see specific associations with interleukin-6. We looked at fractional anisotropy. It tells you how freely water can diffuse along the tract, and this tells you something about the maturity, the integrity of the tract. And a fractional anisotropy is lower, it means that the tract is less mature because water can flow more freely. And this is what we saw here bilaterally along the tract, and it is specifically in areas along the tract that are close to the amygdala, which we thought was quite interesting. So higher interleukin-6 during pregnancy was associated with lower maturity of these tracts bilaterally. We then, unfortunately, only had a small cohort, a small subsample where we had an additional scan at 1 year age. I would say in our current studies, we have gotten quite a bit better in obtaining these scans, but it is quite a bit more difficult to get the scans in the 1 and 2 year olds compared to the neonates, because we acquire all the data during natural sleep, and the neonates just sleep more easily. But here we have a small subsample, and what I want to show you is that if we had just looked at these kids when they were 12 months old, we wouldn't have been able to differentiate those whose mothers had higher or lower interleukin-6 concentrations, because the children actually caught up in terms of their maturity. You would almost even think that maybe they have now an accelerated trajectory. We unfortunately weren't able to look at them a little bit later, but this is what these lines might suggest. So you could say, well, then it potentially doesn't matter where they start, if then they end up fine at 12-month age. But what was really interesting was that it was actually this change during the first 12 months. So this accelerated maturation that then predicted the cognitive development at 12-month age. So this tells us that it's not just a matter of, well, if they get there eventually, it's fine. It's also a matter of how they get there and how this maturational trajectory occurs in terms of later cognitive function. Because for all of these neonatal phenotypes that I just mentioned, we did see associations with cognitive outcomes, here response inhibition, working memory, and cognitive development. We wanted to look at these children when they were 4.5 years age with a measure of general intelligence, so general cognitive ability, and we picked fluid intelligence to see whether we actually at this age see an association between IL-6 and cognitive performance. And this was indeed the case. So higher interleukin-6 during pregnancy was associated with lower fluid intelligence between 4 and 5 years age. And we also were able to identify some differences in cortical volumes that mediated this association, and this was specifically mediated by the pars triangularis volume that was different in these children whose mothers had higher interleukin-6 concentrations during pregnancy. So there's more and more human but also non-human primate work really showing that variation in maternal interleukin-6 concentrations during pregnancy are associated with neonatal brain outcomes. There's a recent study also in individuals 45 years later where serum samples could be analyzed from during pregnancy. Then also there's an interesting study from Finland looking at neurodevelopmental delay in a very, very large registry, and so also some very interesting non-human primate work showing the consequences of higher interleukin-6 concentrations. So to summarize, higher maternal interleukin-6 concentrations during pregnancy was associated with larger newborn right amygdala volumes, functional amygdala connectivity, and functional connectivity within and between multiple brain circuits, especially in the subcortical salience and dorsal attention and visual networks, and with trajectories of white matter development. And we saw implications for cognitive functions in infancy, and also with lower fluid intelligence at preschool age. So we really do have evidence for these various prenatal conditions to be associated with different aspects of fetal brain development. I'd now like to take a little bit of time before we enter the discussion to talk about not only prenatal conditions, prenatal stress, but preconceptional stress. So stress that the mother brings to her pregnancy, stress that she experienced long before she became pregnant. And I want to specifically talk about the intergenerational transmission of maternal childhood trauma. So childhood trauma is a condition that's really highly prevalent. It's almost like about every third person experiences some type of abuse or neglect in childhood. And depending on chronicity and severity, childhood trauma is associated or has been shown to be associated with altered endocrine, but also immune function, higher risk for psychiatric disorders, more obesity, more substance use, earlier mortality, etc., etc. There's really, it's a huge risk factor for many health outcomes. And what is becoming more and more clear is that this risk seems to get transmitted into the next generation, because what has been shown is that maternal childhood trauma is associated with a higher risk for autism spectrum disorders, behavioral problems, perinatal complications. And this can even occur if the children do not experience any kind of abuse themselves. So unfortunately, it is known that there's this vicious cycle of abuse. So sometimes parents who have been abused become abusive parents themselves. But even without this additional risk factor, you can see these higher risk profiles in these offspring. And I'm going to talk about maternal childhood trauma because I'm very interested in potential mechanisms of transmission during pregnancy, but it has been shown that also paternal childhood trauma can increase risk for health outcomes in the offspring. But there it's different mechanisms. It's likely epigenetic changes in the germline that actually gets transmitted to the fetus. So the question is, how can this transmission occur? Most studies are really rather focusing on postnatal processes, because we know that maternal childhood trauma increases the risk for depression in the postpartum period. This might affect parenting behavior and lead to reduced maternal sensitivity in the postnatal period, all risk factors for psychopathology. But we believe that this transmission happens already earlier during prenatal life, because the neurobiological alterations that have been shown in association with childhood trauma include changes in endocrine and immune function, for example, these kind of biological processes that I have just talked about as cues for the fetus about maternal stress and biological mechanisms that can alter embryonic and fetal brain development. So here's a framework, how we're thinking about it, how the various sequelae of childhood trauma that has been established in the literature may lead to changes in maternal biological state during pregnancy and affect the fetal brain, and also potentially affect maternal parenting behavior. So even the postnatal effects that we assume play a role might already get primed by prenatal biological processes. And then there's also the potential for germline epigenetic transmission, so actual epigenetic inheritance of epigenetic changes that have gotten established in the parents, in the mothers and the fathers, and that might persist after conception and get transmitted to the fetus. This for the father, for the paternal line, has been shown in several animal studies that this actually occurs. So I told you there's quite a bit of evidence showing that there are endocrine and immune phenotypes altered in association with childhood trauma in the non-pregnant state. There's a good literature, especially for inflammation. But as you can see here, the evidence is also accumulating in the pregnancy state, showing that maternal childhood trauma is associated with differences in cortisol concentrations and diurnal profiles and hair cortisol concentrations. There's more and more evidence for higher inflammatory profiles in association with childhood trauma during pregnancy. Then I told you about CRH in the placenta. We have shown, and colleagues of us have also shown, that CRH is increasing more rapidly during pregnancy in women exposed to childhood trauma. And then also we have shown that thyroid hormones might be altered in these women as well. And thyroid hormones play a very important role in fetal brain development, especially in early pregnancy. The fetus is completely dependent on a supply by the mother with thyroid hormones. So all these mechanisms could potentially be responsible for already an transmission that occurs during pregnancy. And we wanted to see whether in the neonatal brain, we already see that there are alterations in brain anatomy. And that is exactly what we could show. So we were able to show that maternal childhood trauma was associated with reduced brain volumes in the newborns of women exposed to childhood trauma. This is this gray bar, and you can see that this is mainly driven by reductions in gray matter volume. And when we try to look at whether there are certain areas of the brain that are more affected than others, it was suggested that this was really kind of global effects, global reductions in brain volume in these newborns. As I said earlier, we are always really interested in whether these neonatal phenotypes, neurophenotypes, predict any later function. And so we looked at cognitive performance and executive function at 2 years age and at 4 to 5 years age. And we were again not able to show any direct effects, any associations, but we were again able to show that there is an interaction with a postnatal environmental variable, and in this case, maternal sensitivity. So what you can see here is that those children with a larger brain volume at birth benefit more from maternal sensitivity in terms of their cognitive function than those children with a smaller neonatal brain volume. And this is, we see similar patterns at 2 years and at 4 to 5 years age and across various cognitive domains, which I think is really interesting because it really tells you something about that potentially it's, I mean, we know the brain stays plastic in the postnatal period, and this really gives a lot of potential for interventions, especially in early life when there's such high plasticity, but it seems like there's potentially inter-individual differences in plasticity as well, which I think is very interesting. Okay, so maternal childhood trauma associated newborn brain anatomy and connectivity, and its implication for infant and social emotional behavior suggest intrauterine transmission pathways, we believe that maternal placental fetal stress biology plays a role in this transmission. So it is really important that we understand these mechanisms better to break the cycle of intergenerational transmission as early as possible and develop new targets for intervention. And this is really the aim of this line of work, to kind of try to prevent this early biological embedding of these early experiences. Because it is easier to build strong children than to repair broken men. So the more we know about the earlier experiences and the better we understand how we might be able to intervene, the better. So I think I went a little bit too long. I'm sorry about that, but I'd like to thank my collaborators that I've been working with, especially my colleagues Sonja Entringer and Patrick Watwer at UC Irvine, and Damien Fair and Alice Graham, who I've been doing all the functional imaging work with, and Martin Steiner and John Gilmore at UNC, who I've been also doing a lot of my infant neuroimaging work with. And thank you very much for your attention.
Thank you so much. Amazing as expected. Maisha, if you want to facilitate a couple of questions still before we move into the trainee speakers.
Sure thing. Yeah, not a problem. Should I just read from the chat box if there are any, or maybe if anyone has any questions, also feel free to unmute yourself and turn your camera on and just ask Claudia away as well. But if you'd like me to read it out from the chat box, that's fine as well. So maybe we'll give it a few minutes.
I definitely don't mind getting the ball rolling on this. I have a lot of questions. So something that you touched on a bit, and something that we've been thinking a lot about in our work, is the interaction between the intervention that, especially in the animal models, that we really anticipate studying, and how that impacts maternal behavior, maternal care, maybe in your work even more importantly, how it impacts it during pregnancy and in the postnatal period, especially with some of the later studies. So I was wondering if you'd talk a little bit more about if you account for that, or how you control for it, or what kind of questions you ask your participants regarding that.
In the postnatal period, you mean? In terms of maternal behavior,
or if there's a way that you assess even maternal behavior during pregnancy, if they're extremely high stress, how that impacts the way that they think about or approach their pregnancy, or their lifestyle factors.
I mean, since our focus is really stress, this is why we do a pretty comprehensive assessment of stress. We do this with standard questionnaires, but we also do this with ambulatory momentary assessments of stress in their natural environment. We do this in each trimester over a 40-day period to really also look at variation in stress. We haven't even started analyzing that data yet, but we believe that if there's a lot of variation, that might be really important, or to really look at how often is the woman above her own mean, because maybe it's not even as informative to look at averages across women and then say, okay, women X is higher than women Y, but maybe it's more important when and how often women are above their own mean. So this is something we are really interested in, and apart from that, I mean, we have all the standard questionnaires. We also look at things that might buffer their stress, like ways of coping, social support, and things like that. I don't know. Does that answer your question?
Yeah, absolutely. That's really, I'm really excited about all of those. I look forward to seeing the results.
I have a question, if I can go ahead.
Sure.
Can you hear me? I first
Yes.
want to say a really, really interesting talk. Thanks so much for speaking to us today. I have a few questions, but I'll start with one. I was wondering if you can speak more about neonatal brain volume as a potential plasticity marker and the implications of that. And then my second question was, you showed a graph about the maturation of the track between, trying to remember now, but basically,
Yeah.
Yeah, there was a catch-up group, and you said it's not only, at 1 year they caught up, but they still had like implications for cognitive development, and so maybe I missed it, but if you could explain why the acceleration or the rapidity of that development is significant and might have negative potentially implications for development.
So, so regarding your first question, so I think in general the well-known concept and what we all know about brain development is that the brain stays very, very plastic in the postnatal period, right? And this is why infants and children learn so much faster than adults do because of this high plasticity. But what might be the case is that there is inter-individual variability in this plasticity, and maybe a reduced plasticity could be beneficial if the fetus expects an adverse environment. Maybe from an evolutionary perspective, it would be beneficial to be less plastic and less responsive to the environment, because it could mean that not only might positive influences not have like such positive effects, but maybe also negative effects could harm them less. So this is like an idea that we have, and we found it pretty intriguing in these studies that we saw like a pretty significant interaction with these environmental factors. So environmental enrichment on the one hand, and then also maternal sensitivity on the other hand. So these are just some ideas we have regarding this, and that potentially there are certain like neurophenotypes that might indicate differences in plasticity. And then your other question was why might it be negative for cognitive development if there's accelerated growth? So I mean, this is something that that has been shown, that, for example, in the autism literature, that there is delayed maturation early on, and then there is like a brain overgrowth in early infancy. It's a very common pattern that is observed in autism, and basically it basically indicates things are not developing the way they are supposed to, in the pace they are supposed to develop. But so this is actually why it is sometimes really, really difficult to make predictions of what is a potential predictor for some cognitive and also mental health outcomes, because we don't know, could something that looks like more mature, could that also indicate that there has been accelerated maturation, and it could actually be negative, and might it be like a U-shaped function and not so much a linear function, because like too fast of a growth might also be negative, but also delayed maturation might be negative. And I really believe it's probably both ends of the curve. So there is an appropriate pace that things should be developing in synchrony, and there could be negative consequences if it's delayed, but also if this is happening too quickly, because then maybe not enough attention to detail is paid. I mean, if we just yeah.
Thank you so much for your answers. That was really, really interesting. Thank you.
Okay.
Thanks so much, Claudia. What a wonderful talk. What an incredible amount of data from, spanning so many important parts of the lifespan and so many different assays and ideas. So Just always wonderful to see this work.